Hello and welcome to the final video of this unit. Today what we're going to be discussing is meiosis. So we already talked about how mitosis goes ahead and separates and duplicates body cells. But this time what we're going to be discussing is the process of meiosis, or going ahead and creating sex cells or gametes. So meiosis is part of sexual reproduction, requiring both two parents. So meiosis involves two divisions. We go ahead and start with one diploid germ cell and end with 4 haploid gamete daughter cells. Each cell has a different variation that's unique from either individual parent. So during meiosis 1, we go ahead and start off and split our homologous pairs of chromosomes. Now remember, homologous chromosomes are 2 chromosomes or 2 variations for the same trait. Whereas meiosis 2, we're then going to go ahead and split apart our separate sister chromatids. So we're going to be going through both of these processes and documenting them as we go. So, naming the phases is quite easy. As long as you know the phases of mitosis, you also know the phases of meiosis. So we start off with interphase, but then we move to prophase 1, metaphase 1, anaphase 1, and telophase 1, followed by our first round of cytokinesis. So this is all associated with what we call meiosis 1. After that, we go through the process of meiosis 2. So during meiosis 2, we have prophase 2, metaphase 2, anaphase 2, and telophase 2, ultimately ending with the second round of cytokinesis giving us 4 unique haploid daughter cells. So let's start off with prophase 1. Now, by the way, this all happens after interphase. So remember, interphase, that has the G1, the gap, the S, the synthesis or replication of DNA phase, and also the G2 or the growth phase, where the cell goes ahead and doubles everything inside of it. So with prophase 1, the nuclear envelope disintegrates. So like we see here, the nucleus is starting to go ahead and break apart. Now from there, once again, our centrioles go ahead and start to migrate and produce spindle fibers. So here are our centrioles migrating to different points of the cell, and they're starting to go ahead and produce their spindle fibers. From here, our homologous chromosomes go ahead and pair up. So as you can see here, we have 2 separate sets of chromosomes for the same trait. As you can see, they're right there connected with each other. So these could be things like hair color, eye color, or any variation of different traits. Now at this point, homologous chromosomes can go ahead and do something called crossing over. We'll go ahead and discuss what that means here on the next slide. So just remember, prophase 1, nuclear envelope disintegrates and homologous chromosomes are pairing up. So during prophase 1, we also go through something called crossing over. Now considering that both of these chromosomes are for the same trait, they have the same information, what they can do is swap different parts to create more variations. So here, we have an exchange of segments, by the way, this is the definition for crossing over, an exchange of segments between homologous chromosomes to create variation. So, if we take a look, we can see that we have chromosome A, we have the pink chromosome, and we have the purple chromosome. Now, these are both for the same trait. So let's say that this is for, let's just go nuts. Let's say that this is for red hair, this is for blue hair. Why not? So what they're doing here, since they're both for hair color, what they're actually doing here is they are swapping different parts. So here, now we have one variation for red. Say this chromatid right here might be more of a, let's go with a pink. This one could be more of a purple. And then this one would be more of a blue. So what they're doing here is creating more variation by going ahead and exchanging different parts of the homologous chromosomes. Up next is metaphase 1. So here, our homologous chromosomes go ahead and line up on the middle of the metaphase plate. So they line up on the middle of the cell. And once again, our spindle fibers go ahead and attach themselves to the center of our chromosomes. Remember, that is the centromere. So up next, during anaphase 1, our homologous pairs are going ahead and being pulled apart to opposite ends or different ends of the cell. Now this is done at completely random, so if you go ahead and take a look, Light blue didn't go with light blue, and dark blue didn't go with dark blue. So what we have here is this is random separation, which is a good thing because, once again, the whole purpose of meiosis is we want as many variations as possible. From here, we enter telophase 1 and the first round of cytokinesis. So the chromosomes are no longer homologous pairs, because as you can see, we separated them, and they start to prepare for meiosis 2. The cytoplasm will then go ahead and split into the first 2 of 4 daughter cells. So this ends meiosis 1. So at the end of meiosis 1, our cells are now haploid. Remember, haploid containing half the information. So 2 haploid daughter cells then go through meiosis 2. So in meiosis 2, the 2 haploid cells will then separate sister chromatids to make 4 unique gametes. So the best way to think of this is imagine it as haploid mitosis. So we're going to go through and do exactly what we did in mitosis, but this time we're working with 2 different cells. And at this point, they're also both haploid. So let's go ahead and get to that. So first we enter prophase 2. So prophase 2, our new sets of centrioles begin to migrate and produce spindle fibers. And this is just like it is in mitosis. Also, just a heads up, an easy way to go ahead and distinguish meiosis 1 from meiosis 2 is meiosis 2 always deals with 2 cells. So we have one here and one here. So meiosis 2 deals with 2 cells. So prophase 2, our centrioles migrate and produce spindle fibers. During metaphase 2, the chromosomes line up along the metaphase plate, and our spindle fibers go ahead and attach to the centromeres of our chromosomes. During anaphase 2, the sister chromatids are pulled apart to opposite poles, just like they were in mitosis. And finally, we enter telophase 2 and cytokinesis, our final round. So the nuclear envelope will then go ahead and reform around the daughter cells before dividing into 4 separate cells. Now, each of the 4 daughter cells is a brand new gamete. So remember, the gametes, these are the sex cells, the sperm and the egg, with genetic information different from either original parent. So if you take a look, what we have here, no 2 of these cells are the same. So this one is different from this one, which is different from this one, which is different from this one. So meiosis, we want as many variations as possible. And this helps go ahead and promote that. The final act of all this, or what this ultimately leads to, is the act of fertilization. So we've gone ahead and created the gametes, or the sex cells, so the sperm and the egg. So with fertilization, the male and female gametes unite. So the fusion of 2 haploid nuclei produces 1 diploid nucleus, which makes sense because if you think about it, we have the sperm, which has 23 chromosomes, and we have the egg, which also has 23 chromosomes. And if we go ahead and add this together, that will go ahead and give us our 46 chromosomes. And that would be the first cell that will eventually go ahead and become a human. So the egg plus the sperm give us a zygote. So zygote is a new life in 1 diploid cell. From there, the cell will go through mitosis over and over and over again until it eventually becomes what we know as an organism. So, the most important thing here is we want variation in a species. The more variation one has, the better chances of survival that species is going to occur. The better chances of survival that species is going to have. So meiosis and sexual reproduction guarantee variation and diversity. And it does this in 3 ways. The first is through independent assortment. So the homologous chromosomes separate at random during anaphase 1. Crossing over, the homologous pairs exchange gene segments, creating more variation. And finally, random fertilization. Which sperm meets which egg? It's all a game of chance. So that right there is going to go ahead and close up shop on the act of meiosis. I hope you went ahead and realized a few things, the differences between mitosis and meiosis, and if you have any questions, please go ahead and contact your instructor. They will help guide you in the right direction. So once again, thank you very much for your time. I hope you learned something today, and until the next video, I will see you later. Take care.